First, about cell specialization. So similar to people's cells mature and get jobs as they grow older. Unspecialized and what we consider immature cells are called stem cells, which you've probably heard before. They have the potential to specialize or differentiate, which is the biological term, into various cell types which carry out specific functions in our body. There are embryonic stem cells, which are only found in embryos, obviously, and adult stem cells, which are in adults, right? And these two different types of stem cells have different capacities and capabilities, specifically for differentiation. There are three capacities stem cells can have. Firstly is totipotent stem cells. They can differentiate into any cell type, including extraembryonic and placental cells. These are only really found in early zygotes, right before you even form an embryo, because these can form the placenta and the extraembryonic cells. They're not found inside the embryo, so they could be also found outside the embryo. So this is found only in really zygotes in the very beginning of development of humans. Next, pluripotent stem cells can differentiate into any cell type found inside our bodies. This excludes all of these extraembryonic, placental cells and all of those, but it can turn into any other cell type that you can find in your body. Again, like blood cells, brain cells, liver, kidney, whatever, name it. These are found mainly in embryos prior to birth. However, with technology, it is possible to turn differentiated adult stem cells into iPSC or induced pluripotent stem cells. So you can actually turn an adult cell or like a specialized cell, like a liver cell, back into a pluripotent stem cell using technology. And that's what the I, induced, stands for, right? And the opposite of that, the natural version of that, is called embryonic pluripotent stem cells, which are obviously found in the embryo stage. Next is multipotent stem cells. They can become more than one cell type, but are much more limited than pluripotent stem cells. So they can't differentiate into anything, but they can differentiate into multiple things based on where they are and what they're programmed to do. So these are what most adult stem cells are. So talking about adult stem cells, they're much more limited in their capacities than embryonic or induced pluripotent stem cells. They're only really used to replenish cells, which are made very frequently. Most common example are blood cells made in the bone marrow. So the bone marrow has a lot of adult stem cells, and I'll show you in the next slide a picture of everything that these stem cells can differentiate into, all of the different progenitors, but it's basically all of the different blood cells that you have in your body. You have many, as you know, you have many different components of blood, and there are many different blood cell types. Other examples can include skin, brain, reproductive organs. These make also a lot of, they replenish a lot, so they also have a lot of stem cells, as well as other organs as well, like your colon and other organs also have stem cells buried deep within that replenish the cell layers. Okay, so the multipotent stem cell in the bone marrow can differentiate into many different blood cell types, including macrophages, erythrocytes, which are red blood cells, granulocytes like neutrophils, basophils, eosinophils, T cells, B cells, and natural killer cells. Those are your immune system cells. Okay, you can see a picture right here. So the multipotent stem cell is right here. Oh, sorry, it's black. Here, I'll make it red. Right here, and it can differentiate into these two types, right? The primitive progenitor cells, and again, these can also differentiate into different things, right? As you can see here, but it goes to this committed precursor cell type, and this is where it kind of is like it's committed to going down this one line of specialization. So, for example, if you want to think of it in humans, this is like the kid. These are like the kid, and he grows into a teenager. He can turn into this type of teenager, this type of teenager, depending on what he's raised in, what environment he's raised in, like what these factors are. You can see use these factors, IL-7. I'm sorry, it's clear. So these different factors, based on these, he can determine what kind of teenager he turns into. And this committed precursors, like if he goes to college and he decides to major in like pre-med or biology or something, he's committed down this line now. So he's sticking to this line. He's still not, for example, he's still not a doctor yet, but he's committed going down this line. And then this again is caused by different factors and different various factors in life. Okay. And then you continue down, and you get to lineage committed cells. And this is like, for example, again, he decided that, let's say he's in med school now, so he's committed to this specific career path, and he's committed to this specific pathway for sure. He's just not exactly there yet. So, but he is like one step away, and that would be going to these megakaryocytes, which get broken down into platelets, or these erythrocyte progenitors go to erythrocytes, red blood cells, like I mentioned. This one, monocytes, go into obviously the monocytes like macrophages, dendritic cells. Granulocytes goes into basophils, neutrophils, or eosinophils. This one goes into T cells, natural killer cells, B cells. Okay. And again, these are just different factors. All of these are different factors that would cause it to go down that pathway. Okay. And you don't have to memorize this chart. May be useful to know it like very generally, not actually just for stem cells, but for blood. And you should know what most of these cell types do. You should know what like T cells, natural killer cells, and B cells do, how they're related to the immune system. Obviously, you should know what red blood cells are, platelets are, loosely. These granulocytes and monocytes also very loosely, nothing specific, I think, for them in terms of like blood, their role in the blood. Okay, so what is the mechanism of this specialization? So during development, cells receive orders to differentiate due to various differentiation factors, which tell them what to turn into. Again, like I mentioned, like the chemicals and stuff. There could be other factors. There could be environmental factors. There could be chemicals, biochemical signals. These are not fully researched yet, and the exact details are way beyond the scope of the MCAT, so you don't have to know them, but just know they, based on differentiation factors, they turn into different things as ordered, essentially. When these stem cells reproduce, they undergo something called asymmetric cell division, which is kind of important. It's a form of mitosis, but the product of asymmetric cell division is two daughter cells which are not identical. This is because one daughter cell remains in like the stem cell like has stem cell like features, so it can continue making new cells. So it's essentially like the spawning point, right? Whereas the other one will be the appropriate cell type. So whatever cell type you're making, like a liver cell or something like that, and it will lose its stem cell properties, so it won't be able to reproduce or specialize anymore. But it'll be able to it'll gain all of the functions of the cell type that it's meant to be, like the liver cell function. Okay, so it makes, here I can draw a picture just to illustrate. So you have a red stem cell. It undergoes asymmetric cell division, and you end up having, make this blue actually, and you end up having two children. One is, so I'll draw like this. One is the liver cell, and it has all the functions that a liver cell would have, right? But it doesn't have the functions that stem cells have. It loses that function, and another would continue to be a stem cell, right? And this one would, again, split more, and then so on and so forth, like that. Essentially, it'll continue going that way, right? And that's how you make more liver cells while also kind of keeping your spawn point set to spawn more of those cell types, essentially. Okay. In adult stem cells, this happens via signaling cascades and cytoplasmic factors, which allow for this asymmetric division. You don't really have to know the specifics and the details. Those are, you might have learned that in your cell biology class if you took one in undergrad, but it's not really that important for the MCAT. Just know that there are a signaling cascade and cytoplasmic factors which tell one to remain the way it is, another one to change. In adult stem cells, this process is slightly different. It's regulated via epigenetic mechanisms, which control gene expression in the stem cell differentiation. Again, remember epigenetic is so all of genes in every cell, your liver, your brain, your heart, your blood, everything has the same DNA. The reason it performs different functions is through gene expression. You only express liver-based functions in liver cells, and like brain functions in your brain, obviously, without expressing your liver function. So the way it does that is through epigenetics. So things attach onto the DNA or control the DNA somehow, so that it doesn't get affected. So essentially really latches onto the DNA, or it alters like the shape of the DNA in some way, like condense it, uncondense it. That's your methylation, acetylation, those types of things. Control which genes get expressed in that. And that's how you control stem cell differentiation via epigenetic factors in adult stem cells. Okay, next let's talk about cell migration. So during the embryonic stage, prior to differentiation of pluripotent stem cells, the cells migrate in response to chemical signals to form the three primary cell layers: the ectoderm, mesoderm, and endoderm. And I put here what they are: the outer layer, middle layer, and the inner layer. Once these cells in these different layers differentiate into appropriate organ cells, they also migrate to form the organ morphology. So it continues with migration. And so, for example, the ectoderm would be things like your skin and those like outer organs. Mostly the mesoderm would be your kind of like middle organs, and the endoderm would be like inner organs. And you can look up, if you would like, you can look up a list of ectoderm, which organs come from the ectoderm, which from the meso, which from the endo. I don't think it's very important, but if you want to, in case just for your own knowledge, please feel free to look that up. And again, once they differentiate into their appropriate organ cells, they migrate to form the organ morphology. So, for example, how does your hand form like these like finger things, right? Well, your ectoderm has to make like skin cells and bone cells, then it has to migrate to this area, and then it like forms these specific shapes, right? And then it gets rid of any other shape, like it like literally like cuts it out like out of like marble, like chisel. You can chisel it out of marble. Essentially, that happens. It just removes everything that's not in the right shape. Same thing happens with like your liver. It forms